Hi, my name is Catherine Morrison, and I'm here to do a presentation in fulfillment of the requirements of the curricula for Chem 530, Concepts of Biochemistry. The instructor is Dr. Sean Stacy and is due July 15th, 2026. So, the presentation today, we trace how the cell controls two opposing pathways. This is glycolysis, which breaks glucose down to release energy, and gluconeogenesis, which builds glucose back up. Our recurring character is the enzyme aldolase, and especially its liver form, aldolase B. By the end, you will see how one reversible reaction ties together: metabolism, gene regulation, and human disease. So let's get started. Here are the objectives for the presentation. First, we will follow glucose from the plate into the cell. Then we will walk through glycolysis and gluconeogenesis and see how they are reciprocally regulated. We then will zoom in on aldolase, its mechanisms, its tissue-specific isozymes, and how the liver switches on aldolase B. Finally, we connect all of that to a clinical case, hereditary fructose intolerance. First, let's find out how glucose is acquired in the body. Well, we eat, and food is made out of majority of carbohydrates that are ingested. Here's a picture of an outline of the human body and the major parts that are used in digestion. The chewed up food goes down into the stomach, and then from the stomach, it goes into the small intestine. In the small intestine, some enzymes work on it and are absorbed through the small intestines. Then anything else will go in through to the liver through its hepatic portal. And in the liver, we get disaccharides that were broken down in the small intestine. And in the liver, they are broken down even more into monosaccharides, which is the glucose, galactose, fructose, etc. From here, this glucose will be released into the bloodstream, and then from the bloodstream, the glycolysis will go into the cell, and it begins in the cytoplasm of the cell. So here is a pathway of the full glycolytic pathway. Glucose goes down to pyruvate in 10 steps with enzymes labeled on each arrow. Do not try to memorize every intermediate. Focus on the 3 orange steps: hexokinase, PFK1, and pyruvate kinase. Those are irreversible regulated control points, and PFK1 is the main gatekeeper. So here we have hexokinase and glucokinase. We have hot phosphoglucose isomerase. We have PFK1. We have the aldolase. And then we have the glycoaldehyde 3-P dehydrogenase. Then we have the phosphoglycerate kinase, phosphoglycerate mutase, enolase, and then pyruvate kinase. If you notice, the 3 ones are in the first one, which is the glycose/glucokinase. And the glucokinase is usually used in times of fasting or heavily body stress, but normally it's the hexokinase that will go from glucose to glucose 6-phosphate. And then the other next one is the PFK1, which breaks down fructose 6-phosphate and fructose 1,6-biphosphate. This is an irreversible pathway, just like hexokinase, means it only goes one way. The other steps can go either way, up or down. Then the last one will, of course, is the pyruvate kinase, which will break down PEP into pyruvate. Those are the more important steps in the whole pathway. And during this time, you will see where energy is made and used. First step is a hexokinase where the ATP is phosphorylated to the ADP. And then the next one, where the orange irreversible, we use the ATP again that goes to ADP. And then in the 2, which is a GHAP and a dihydroxyacetone phosphate. In these ones, you can make 2 for 1, but it does use NADH and it yields NADH plus phosphorus. And then we go to the next one, the 1,3 biphosphoglycerate. When it's broken down by its phosphoglycerase kinase, it will use ADP to produce ATP. And then the last one, pyruvate kinase, will also convert ADP into ATP, which is hydrogen, and this is into pyruvate. Now, all of this is happening within the cytoplasm of the cell. Next process that we are going to talk about is gluconeogenesis, which is essentially glycolysis run backward. Pyruvate up to glucose, 7 of the steps simply reuse the reversible glycolytic enzymes, but there are 3 irreversible steps that cannot run backward, so dedicated bypass enzymes handle them. As we can see, there's going to be some orange steps, and these are irreversible bypass reactions unique to gluconeogenesis, and they circumvent the 3 irreversible steps of glycolysis. Now, if you remember when we were talking about glycolysis, there were 3 steps, the hexokinase, the PFK1, and the pyruvate kinase, that they were irreversible steps, and the other steps were reversible. The same thing happens here. We go from pyruvate carboxylase, which happens in the mitochondria, which breaks down the pyruvate into oxoacetate. And then we have the PEP carboxylate kinase, which is the PEP-CK. And that breaks down the oxaloacetate to phosphoenolpyruvate. And then we have the enolase, which breaks it down to 2-phosphoglycerate. The phosphoglycerate mutase, which breaks that down to the 3-phosphoglycerate. The phosphoglycerate kinase, which breaks down that to the 1,3-biphosphoglycerate. And then we have the glycerolide 3-P dehydrogenase. And if you notice and remember that in the previous slide, we had a breakdown to glycerol aldehyde 3-phosphate and dihydroxyacetone phosphate. The same thing occurs here, but we're going from 1,3-biphosphoglycerate to the 2 enzymes. And then we have the aldolase, which also breaks down the glycerolide 3-phosphate into dihydroxyacetoacetate phosphate. And then they break down to the fructose 1,6-biphosphate. Then we have another irreversible reaction, which is a fructose 1,6-biphosphatase. And this breaks it down to fructose 6-phosphate. And then we have the phosphoglucose isomerase that breaks it down to glucose 6-phosphate. And then we have the glucose 6-phosphate, which occurs in the ER and liver, and this is converted into glucose. So now let's put the two pathways side by side. The key idea is reciprocal control. The same signals that turn one pathway on turn the other off. So the cell never runs both at once and wastes ATP in a futile cycle. The master switch is fructose 2,6-biphosphate, high after a meal under insulin to drive glycolysis, and low during fasting under glucagon to favor gluconeogenesis. AMP and citrate fine-tune the balance according to the cell's energy charge. And as you can see, the net reaction is glycolysis is a glucose to 2-pyruvate, which yields a 2-ATP and 2-NADH. The glucogenesis costs 6 ATP equivalents, so it goes from 2-pyruvate to glucose. Then we have the committed one-way enzymes, which is in glycolysis, it's hexokinase, glycokinase, PFK1, and pyruvate kinase. In gluconeogenesis, it's glucose 6-phosphatase, fructose 1,6-bisphosphatase, PEPCK, and pyruvate carboxylase. They're turned on. The glycolysis is insulin, which after we've eaten, a high AMP, fructose 2,6-bisphosphate, fructose 1,6-biphosphate. And then in gluconeogenesis, it's glucagon, which is fasting, acetyl-CoA, citrate, and cortisol. It gets turned off by, in glycolysis, ATP, citrate, glucagon, low fructose 2,6-biphosphate. And then gluconeogenesis, it's turned off by AMP, fructose 2,6-biphosphate, and insulin. And if you notice, some things that are turned on will turn off the other one, and vice versa. And they dominate when the glycolysis occurs more often when you're fed, when your blood glucose is high. And then your gluconeogenesis, it comes in when you're fasting, starvation, or prolonged exercise. And you must ask, why can they not run at the same time? The same signals switch one pathway on while shutting the opposing enzyme off. So cells never waste ATP by running both, which is a futile cycle. There is a master switch, which is fructose 2,6-biphosphate, and it's high after a meal and causes insulin to kick in, which activates the PFK1 in glycolysis and blocks fructose 1,6-biphosphate, which occurs in gluconeogenesis. And this is during fasting and glucagon. And pKa lowers it and reverses the balance. Also, AMP and citrate acetyl-CoA fine-tune it. AMP favors glycolysis, while citrate and acetyl-CoA favor gluconeogenesis. In our next slide, we're going to talk about aldolase, which is a reversible step shared by both pathways. In glycolysis, it cleaves fructose 1,6-phosphate biphosphate into DHAP and G3P. In gluconeogenesis, it runs the exact reverse, joining them back together, and the enzyme does not pick a direction. Substrate levels and pathway flux do. It is a near equilibrium reaction, and the class 1 enzyme works through a lysine shift base, which is in the next slide. In this slide, we're going to be talking about aldolase, its catalytic mechanisms, and isozymes. This is the chemistry. In the class 1 mechanism, an active site lysine attacks the carbonyl to fructose biphosphate to form a shift base. A retroaldo cleaves, then releases G3P and leaves an enzyme-bound alanine, which is repronated and finally hydrolyzed to free DHAP, which is reversible, which is exactly why aldose can run both ways. Note that class 2 enzymes in fungi and bacteria use a zinc ion instead, making them a selective drug target for future use and research. In this slide, we're going to talk about 3 isozymes that tune the same reaction to each tissue. We have aldolase A, which dominates skeletal muscle and red cells, and its level in blood rises after muscle injury. Aldolase B is a liver form and uniquely handles fructose, and aldolase C is found in the brain. And keep aldose B in mind because it drives the rest of the talk. Aldolase B is central to liver metabolism and fructose handling. It is the only isozyme that efficiently cleaves both fructose 1,6-biphosphate and fructose 1-phosphate, which places it in at the crossroads of glucose and fructose metabolism. In everyday metabolism, it runs the reversible triose step in both directions. For dietary fructose, fructokinase makes fructose 1-phosphate, and aldolase B splits it. The committed step of fructose handling, and it loses that activity and you get hereditary fructose intolerance, which we cover in the case study. In this slide, we're going to talk about how the control of the aldolase B is liver specific, and it also is a promoter. So why is aldolase B a liver enzyme? Because a cluster of liver-enriched transcription factors binds its promoter. HNF1, which is also hepatocyte nuclear factor 3, is a pioneer factor that opens the chromatin. HNF1, which is the hepatocyte nuclear factor 1, is the master activator. And C that I/EBP and DBP add metabolic and circadian fine-tuning. They act combinatorially and clinically, losing HNF1 alpha function, as in HNF1A MODY diabetes, lowers hepatic aldolase B, showing how promoter control sets tissue-specific enzyme levels. This slide, aldolase B and metabolic homeostasis, pathways regulation in disease, will integrate everything. Aldolase B sits where metabolic pathways, gene regulation, and tissue expression meet. Pathways, it runs those steps and cleaves fructose 1-phosphate in fructolysis. The regulation part is liver factor sets how much enzyme is made, while substrate levels set which direction it runs. For the expression, it is high in liver, kidney, and intestines, and turned to fed and fasted states. When the balance breaks, you get hereditary fructose intolerance, HNF1A MODY, or a fructose-driven fatty liver. And this is our clinical case slide of hereditary fructose intolerance. Here is a disease in practice. It is an autosomal recessive loss of aldolase B. Infants are symptom-free until weaning. Then fruit, juice, or sucrose triggers vomiting, severe hypoglycemia, and liver problems. The mechanism is a fructose 1-phosphate trap. It accumulates, sequesters phosphate, and depletes ATP, which stalls glucose production. Diagnose with gene testing rather than a risky fructose challenge, and treat with lifelong avoidance of fructose, sucrose, and sorbitol. The key point is that there is no drug, and you simply remove the substrate in the missing enzyme and cannot handle. In this slide, we're going to talk about metabolic states, which include fed, fasting, and exercise. This slide also will demonstrate how hormones set the direction. The insulin to glucagon rate and energy change signals switch glycolysis and gluconeogenesis on and off across the day. Zooming back out to the whole body, hormones set the direction. In the fed state, insulin dominates, glycolysis and storage are on, and fructose 2, 6, biphosphate is high. In fasting, glucagon dominates and the liver makes and exports glucose to the brain. During exercise and starvation, energy change signals such as AMP and epinephrine drive fast ATP. The Cori cycle recycles lactate back to glucose, and prolonged starvation shifts to ketone bodies. It is the same reciprocal control now at the level of the whole organism. The key takeaways to this presentation is to remember: 1. The two pathways share 7 reversible steps and bypass 3. 2. Fructose 2,6-biphosphate is the master switch set by the insulin to glucagon ratio. 3. Aldolase uses a shift-based mechanism with no metal needed. 4. 3 tissue-specific isozymes fit the reaction to each tissue. 5. Liver transcription factors set aldolase B expression. And 6. Break these controls and you get disease. That is the arc from reaction to regulation to disease. I hope you have enjoyed this presentation as much as I have enjoyed presenting it. This is the last slide, and these are sources which primarily come from the textbook Leininger and Harper's biochemistry text, along with the cited figures and web resources. I'm happy to share the slides and will take any questions. Thank you.